So we've been working on this from a bioinformatics perspective for a while, trying to define our set for the CanEuCre project. The first thing we had to do was to develop a pool of candidate genes. These are genes that would have a pattern expression that was relevant to the community. We did that through both analyzing expression data and through working with the local and international communities to find genes and expression patterns of relevance. We then look at those genes and we say we want as simple a problem as we can take on. So we don't want to have multiple genes in the same piece of DNA because we don't want to worry about what those genes are doing. We want to knock out the activity of the one gene that's in C. So we throw out most of the genome because it has too many genes nearby. We don't want to have complexity in terms of the structure of the genes. So many genes have multiple promoters and multiple open reading frames. We want one that has one translation start because we have to put our protein of interest, in this case it's going to be a pre-recombinase, at that start position. And we don't want to worry about what's happening in other places. There's some issues around nonsense-mediated decay, which I'll step over, but they do come into play with this design. And then we are working with a restricted library, so we have to have a back that's available in our library, and so it has to be pulled out of the RP11 system. We then sort of take all that information, develop some bioinformatics procedures to look at the genome annotations, and then refine that down until we get a smaller set. So we started with 3,000 candidate genes. We quickly came down to about 400 genes that we could potentially work from. And then we went back to the community and said, of these 400, which ones would give us the reach that we needed? We curated, for the ones that we were leading on as potential candidates, we curated as much as we could from the literature. We looked at the epigenetic profiles that are available in the ENCODE project, tried to compile all that in place, and come out with the case of the backs that we thought were most likely to contain all the regulatory sequences. Then we do some design, and we pass it off to the laboratory team, and they start modifying the backs. So what happens once we've got the backs is that they do get modified, and we work with a system for studying these, which involves a deletion strain of mice. It's called an HPRT deletion mice. And what we do is we take our back sequence and we add some sequences onto the edge of it that will allow it to have homologous recombination into the deleted region of the HPRT gene on the mouse X chromosome. And then we can select those cells that repair the broken gene by a hat-based selection in cell culture, such that the cells that we get growing out of that cell culture are going to have a repaired gene, and we can go ahead and develop mice to study further. A brief side note, we had an SOF project that we developed having Genome BC support because we wanted to see if we could replace this sort of homologous recombination system with a more popular system called the Gateway system, which is used by Life Technologies, formerly in Vitrogen. And so that involves putting a particular target sequence into the genome and then using the R4 integrase to promote the recombination of your DNA sequence into the site. So we did that. We put it into the HPRT locus just because we could get it in there very easily to see if it would work. And we profiled that. We generated a mice with that line, and our partners at Vitrogen and Life have been working with that and trying to optimize it. But for our CanEuCre project, we found that we were still getting higher efficiencies with the hat selection and the homologous recombination. So for the project that we're doing, we're continuing to use recombination, but it may be that this one comes along in the future. So this is just a slide saying that when we do the recombineering, what we're inserting is a gene here, which is called ICRE, ERT2. And so that's a gene that can be has CRE, that's the CRE gene that we talked about. And then it has the ERT2 sequence, which essentially allows us to activate the CRE activity by exposure to a tamoxifen. So you can control the activity by adding tamoxifen to the system. That sequence can also be, through manipulation, taken out, so you're left with just CRE in the system. And then what we do with that, I don't have the slide in that spot, what we do with that is we introduce that sequence into a mouse that has a LAC-Z gene that gets activated in the presence of CRE. And so that blue staining that you'll see in the brains comes from that LAC-Z gene that's somewhere else in the genome getting turned on by the CRE. So these are the maxi promoter genes that we're working up for this this project. The ones that are remaining, there's always some that get lost along the way, so it gets pruned down all the time. So if your favorite gene's in there, that's good. If it's not, the maxi promoter pipeline is pretty well closed off, just because it takes a while to do this manipulation and work it out. But the good news is the mini promoters pipeline still has some entry points, so if you have a favorite gene or expression pattern, you can still bring it to our attention and we can squeeze it in. So let's talk about going small. So what we want to do on the small side is we want to be able to fit these things in adeno-associated viruses. These are sort of the flavor that's preferred for gene therapy applications. They don't cause any human disease. They don't integrate into the genome, so that you don't have to worry about the negative impacts of inserting a gene somewhere and causing cancers, which is the classic problem with gene therapy. It's good for non-dividing cells, so it doesn't replicate in large enough. So you're not going to have if you put it in dividing cells, it's just going to get lost over time. Now, the key thing that makes this project possible with working with viruses, and the sort of the big breakthrough of the last few years of viruses of this kind, is a strain of AAV called AAV9. And AAV9 has the capacity to get past the blood-brain barrier. It's not quite clear how it does that, but it has the capacity to get through that zone. And so for our purposes, that allows us to add the virus directly to the bloodstream and have it go into the brain, infect cells, and not have to do anything particularly special in order to get it there. And so that allows us to test in vivo the activities patterns that we direct. As I mentioned, non-insertional, and there's no disease associated with the virus. So from a bioinformatics standpoint, what we're going to do is we're going to take a gene. In this case, we'll look at Wyeth G1, your favorite gene 1. And we can see that if we go through our bioinformatics analysis, looking at everything we can put our hands on, doing all our algorithms, we emerge in this case with 11 candidate regulatory sequences and a promoter at the start of the gene that we can capture. And then what we do is we try to design a series of compact DNA sequences that will capture those pieces, and then we can go through and test in either the virus or a plasmid-based system. I'm at 10 minutes, but we're doing okay. So the design process is somewhat similar to maxi promoters at the front end. We have to select genes that have a pattern of expression that we're after. We have to curate the known regulatory regions into a database and take in all of the data that we get our hands on from larger scale projects. We predict the candidate regulatory regions, and then when we've broken it down to those genes that we think have a relatively small number of regions to test, we go through and we review each one in some depth and try to understand what the regulatory sequences might be. So these are the genes that are currently under development in our mini promoter AAV pipeline with regulatory sequences coming from. We have about 50 more slots that we'll be working up over the next 6 months. So if you have a favorite pattern of expression or a favorite gene that you think would be just lovely to have driving an expression in the brain, catch me or Dr. Simpson afterwards to talk about it. Okay, so let's talk about AAV, because this is kind of a new thing for this community. I don't think we have a lot of researchers in town working with adeno-associated virus and doing these types of tests. What we're doing is we start with injecting the virus into the temporal vein at P0, the day of birth. I mentioned that you can traverse the blood-brain barrier. It seems to be particularly effective at getting past the barrier at a young age, like the day of birth. The protocol does reach the entire body, so we're going to look into locations other than the brain in our analysis. And what I'm going to show you in the first pass are ubiquitous promoters, because we are still building the smaller compact promoters for the working project. So our initial tests have used sort of more traditional ubiquitous promoters, and we're going to see how well we're reaching different parts of the body and the brain. So this is the basic system. It's an AAV virus. We put an I3 under a promoter of the virus, and then in the genome, the CRE will promote the removal of a stop codon, and that will result in the expression of the lysis gene in that mouse. So the first thing we have to do is see if we can get the virus across the body, and so it's a bit of a challenge to hit the temporal vein precisely. We've got some technicians who are extremely good at it. So what you'll see here is our mice that have been injected with green food coloring. It's not toxic. And they're fine, they're alive. And so what happens when you hit the temporal vein, it just sort of instantly flushes throughout the body, and the mice turn green entirely. And if you happen to miss the temporal vein, you get a little bulge of food coloring up there at the site of injection. So I think we're pretty darn good at this now, and it goes sort of straightly along the way. So then we started doing some basic things. We said, how much expression are we getting in the brain? Do we replicate what's going on out there? And so we're looking at sort of dose dependence in brain expression. So this is looking 3 weeks after injection, which is kind of the short side of these types of experiments. The virus, this is a CMV viral promoter in the gene therapy virus. And you're just seeing that it is dose dependent. As we increase the dose, we do reach more cells in the brain. And you can see that there's some preferential locations in the brain that are picking up getting hit by the virus. And that there's sort of the closer you are to a blood vessel, the more likely it is that you're going to get hit by the virus. The next set of experiments was to look to see sort of the temporal influence, so whether these promoters how long you have the system going. And we've been told that it was sort of 3 weeks was the lowest day that you want to go, and here you see 8 days, 14 days, and 21 days. And you can see it's building up in terms of the activity. And if you go longer, if you look from 21 to 52 and 84 days out, you can see that you get sort of a broader range of cells that have been hit by the virus and have had a chance to recombine the genome and introduce the flu expression. So we're pretty happy at that point. We're reaching much of the brain successfully. We can deliver the virus past the blood-brain barrier, and leads us to have good hope. So this is going in and looking at a little higher resolution at different locations and seeing the expression pattern. This was at P21. And this here is the spinal cord, so we're reaching the spinal cord as well, which might have some therapeutic potential. So we've got a ways to go yet. We have, by the end of this project, we'll have gone through about 25 maxi promoters and about 35 mini promoters and developed them with this system. And that will be contributing to the Therapies project as well, and they will be absorbing them into their panels. We will have established an AAV promoter toolkit for brain research, and we're underway with that effort. And then what we'll be moving into after that is to see if we can actually deliver therapeutic expression of genes using this system. We have some initial forays on a separate project in that direction. So one of the things that we pride ourselves on is being an open box for what we're doing, and so we try to make everything that we do available to the research community without restriction. So the bioinformatics software that we create for the regulatory sequence analysis is up on our website. Some of it's in other repositories, but there's links to that from there. We keep project data online on Therapies.org. It needs a little refresher, but it's coming. For the constructs, they're available from AdGene. So if you want the DNA constructs that we've been working with, you can obtain them from there. And for the ES cells and the mice that have been produced in the project, they are available from the Jackson Labs and their cell population sources. So there's a brief statement that this is a big team of people participating, and that we are pleased with the interactions that we've had across the project with all involved. So I will stop there.